Hello, and welcome to this ethical dilemma in science, where we're going to take a look at genetically modifying pigs as a source of organs for transplantation. And so the prompt for this came from a CNN health article on CNN's website. Made a lot of news within the last couple days. We're in the first full week of 2022. But basically looking at a situation where we've got a number of people dying every day as a result of a lack of organs for transplantation. And so, in this situation, we're looking at an individual that has terminal heart disease that was approved through FDA, Food and Drug Administration, emergency use authorization to receive a transplant of a genetically modified pig heart. And this has been a pig heart that has been altered in a way to kind of improve the likelihood that it's going to be successful for this individual. So to take a step back, in December 2020, so a little over a year ago, the Food and Drug Administration approved the use of one type of genetically modified pig as a source of food and potential therapeutic uses, potential use in human medicine. And so what we're looking at is this idea that we can go through and modify these cells in a way, and I'll go through this in a little bit more detail in a couple minutes, but decrease the likelihood that they're going to be recognized as foreign or pathogenic or disease causing or damaged within our body, and hopefully allow them to become established within the body and work for kind of a longer period of time. More recently, as I started researching this article in fall 2021, they did some tests. They worked with a patient that had died that was still on life support. They received ethical approval, and they basically attached one of these genetically modified pig kidneys to the individual. And we're able to look at how the kidney was going to be functioning in this environment, and they were able to show kind of a restoration of kidney function and kind of urine output and a bunch of other physiological parameters that we're not going to worry about. But it was evidence that the kidney was working, the pig kidney in this kind of human transplant situation was working properly, and it didn't appear to be being rejected. And so it gave a good first start in this process. Now, animals have been used as a source for organs since about the 1960s. So, basically 60 years now at this point. And so we've used pigs in a variety of ways throughout the history of humanity. We've domesticated them. We've got them as a food source. But more recently, we've been using them in medicine. And so for a patient with diabetes that can be treated with insulin, one of the classic ways of treating diabetes, starting about 100 years ago in the 1920s, was to collect pig pancreas, an organ within our abdominal cavity, within our belly region, that produces insulin. Basically take them from a slaughterhouse, these pancreas, grind them up and isolate insulin that could then be given to a patient with diabetes so that they can control their blood sugar levels. But for many individuals, the pig insulin was slightly different than the human insulin, and it was recognized because of that difference, and their body mounted an immune response to it. So ideally, what you want is to be able to get human insulin. But huge ethical questions. We can't go around and start collecting human pancreas and grinding it up. Huge ethical issues there. And so what we've been able to do using, genetic engineering, starting about, in the 1970s, about 50 years ago now, I think it was first approved in the early 1980s, we've been able to kind of alter bacterial cells so that they can produce human insulin for us. And now at this point, I think all of the forms of insulin that are now available are going to be this recombinant form of human insulin produced in bacterial cells. And so there is this opportunity then to use what we know about genetic modification to improve human medicine. So what is it that's going on? So in this situation, basically we've got two cells here, and in reality, they don't look like this in nature. Don't look like this underneath a microscope slide. but it's kind of representing what we're thinking about. And so we've got a pig cell here over here on the left. We know it's a pig cell because it's got a pig in it. We got the cell boundary kind of around the outside. And then on the surface of all of our cells are going to be a variety of proteins. And if you're not a biology major and you don't want to necessarily know what proteins are or anything like that, they're basically kind of biological molecules, biological shapes. They're going to be found, in a variety of locations within our bodies. But in this case, these pig cells that we're looking at are going to have these pig proteins. We got little piggies kind of growing along the outside of the cell. In contrast, we've got a human cell over here on the right-hand side. We know it's a human cell because we've got the kind of man from the Voyager golden plaque sent into space, 45 years ago now at this point. And so the human cells, like the pig cells, are going to have protein shapes along their surfaces, but the human cells are going to have human shapes along their surfaces where the pig cells had pig shapes along their surfaces. Now, if we were to take this pig cell and transplant it into a human, what we're going to see is that our body's immune system, our body's protective mechanisms are going to go through and try to fight off disease. They're going to try to fight off damaged cells. And so cells that look different from what was normally found within the body are going to be one of two things. They're either going to be associated with a disease or associated with, say a parasite or something that is kind of invading the body to kind of take over it. A tapeworm kind of going through and getting into your digestive system and causing major problems there. And so we've got protective structures within our body, referred to as our immune system. And so we're going to have cells and then little biological kind of proteins that they produce called antibodies that are going to go through. They're going to circulate through the body looking for disease and looking for things that are unusual that could indicate danger in essence to the body. And so this pig cell placed into a human environment, our human immune system, our protective system is going to go through, and we've got all of these little soldiers going through, and they're going to be attacking these pig shapes, these pig proteins on the surface of the cells. So overall, what happens then is our immune system is going to go through and reject this pig cell as being foreign. It's going to reject it as being abnormal, and they're going to attack and kill it. And so, if this is a parasite that is getting into your body, you want to attack it because you don't want the parasite to get established and cause a big disease process that, could potentially kill you. But in this situation, we're interested in taking cells and organs from one animal and transplant them into a human being. But we've got all of these pig proteins, these pig shapes on the surface of the cells that basically say, hey, this cell here isn't normally here. Take a quick look at it, and because we don't have the ability to recognize it's important to have a heart with these protective mechanisms here, they're just looking at, is it normally found within the body? And if it's not normally found within the body, let's get rid of it. So they're going to kill these cells. So what happened with these genetically modified pigs is they went through, and again, back this up a slide, we had the pig proteins, pig shapes on the surface of the pig cells before. Now what we're doing in this genetic modification of these pigs is we're going through and doing a couple of things. The first is that we're going to remove those pig proteins, those pig shapes, off of the surface of the cell. And they went a step further. They actually put in some human genes, some human genetic information in the form of DNA into these cells so that they start to express human shapes. We've got these little human heads that are now showing up on the outer surface of these cells. We've still got our immune system, our protective system kind of circulating through the body. So we've got a little soldier kind of running around here. But he isn't able to look inside of these cells. He's only going to look at the outside of the cells, and the outside of the cells of this genetically modified pig cell with the human heads, human shapes along their surface, are going to look from the outside very, very similar to a normal human cell, which has, again, human heads associated with it. So in this situation, these pigs have been modified in a way that they're less pig-like and more human-like. And in doing so, hopefully minimizing our body's protective response against those cells. So we're not going to attack the transplanted heart not attack the transplanted pig tissues in the human body, and then allow that to, hopefully pump the blood throughout the body for that individual for a long period of time. So that gives rise to our ethical dilemma. Let me move over so we can actually see what's present on the slide. So when does a pig stop being a pig? Should there be limits on how much we're altering pig cells with human genes? Are pigs who are expressing human genes still pigs? Or are they something kind of in between? Do they become almost like a chimera with a mixing and matching of information from two different organisms? And then finally, and a little bit controversial, not doing it to be titillating or cause a huge reaction, but to generate some thoughts to be thought about. Do pigs expressing expressing increased numbers of human genes, do they stop becoming pigs and start becoming maybe not quite human, but potentially subhuman? And what does it mean if we are starting to create organisms in some way that are kind of midway between animals and human? And I recognize humans are animals, and that's a whole separate conversation. But what is it that we're doing? What is it? Are there ethical limits? Are there the rules that need to be in place that determine how much we can alter these animals to make them, better for what it is we need them for? And, before you say, this is far-fetched, and yeah, it is a little bit far-fetched now at this point, the effects of altering animals to become more human is a prevalent theme within science fiction. Going back to the island of Dr. Moreau, published, I should know this, I think it was in the 1890s. So, roughly 130 years ago, late in the 19th century, was talking about kind of altering animals to make them more human-like, to make them, better servants for a Dr. Moreau. More recently, in the movie Rise of the Planet of the Apes, released in 2011, they were looking at testing an Alzheimer's drug, a viral drug which would go through and protect our brain cells from being lost with a degenerative disease, dementia like Alzheimer's. But in that, what they're going to be looking at is that they altered the ape, the chimpanzee that received that, and basically triggered the much more sentient primates, which were the basis for the theme within the Rise of the Planets of the Apes. And so that's it for our ethical dilemma in science for this week. I'm going to post some links below, both on the Canvas site as well as the YouTube site. So if you're interested in reading more about this, I'll link the CNN articles as well, some additional information as well. And then, this production was put together with the OBS Open Broadcast software and the Video Prog Vlogger video editing system. So thank you and have a great week.